On behalf of Arun Ravindran and myself and Mary Jane Esplin, who is leading this mentorship initiative, wanted to welcome you to this iteration of the scientist, clinician, investigator mentorship group. Today we are very fortunate to have Dr. Anthony Levitt, who is a professor in the Department of Psychiatry, who has a research and clinical interest in mood disorders and is the medical director of the Family Navigation Project based out of Sunnybrook, has a long and varied career that we are delighted to hear more about today, and we're grateful to have you here, Dr. Levitt.
Well, definitely my pleasure, and my pleasure to talk about randomized controlled trials, which is the topic today. And if you want to start the slideshow.
Okay.
We can get underway. So I don't usually spend any time on my credentials, but I just wanted to point out at the bottom there that I am one of the chairs of the Randomized Controlled Committee for CIHR. I've been on the RCT committee since 1997, both as a member and for a considerable amount of time as the chair. And so I am going to share today some insights into what doesn't work and what works in terms of making an application for an RCT. And also I'd like to talk about the value of the individual. So I'm going to talk about the joy of clinical trials from N of 1s to 1000s. And I make few apologies for the fact that I'm just going to pick out highlights. This is neither a presentation about N of 1 methodology, nor is it a presentation about all randomized controlled trials, but I want to show the breadth of what's of the joy of doing clinical trials. So here's an overview. Next slide, please. So I'm going to start with the value of N of 1, and then I'm going to talk about some guiding principles for writing successful non-N of 1 or N of greater than 1 RCTs. And I'm going to talk about hypotheses, sample size, and blinding. And I know that sounds incredibly dry and boring, and I tried to make those three things a little bit sexier. Hopefully, it'll be a bit more interesting when I get to that point, but it looks kind of dry. So let's start with the value of N of 1. And you can read about this, and you can get self-help. There are books out there, and there are articles. But the next slide is just a cautionary tale about what happens if you're too much into self-help. So I'm going to give you some help. Next slide. So this is really about N of 1 methodology, about the power of an individual to make change in the future of mankind. Next slide. And I'm going to talk about why we do N of 1s, what's the problem with the randomized controlled trials, which I know is I shouldn't be saying that if I'm the chair of that committee, but there are problems. What is an N of 1 trial, how to apply it, a brief focus on both drug studies and sequential N of 1s, and then my plan for world domination. Next slide. Okay, so this is a quote that comes from Rohr. It's from 1983. It's as relevant then and now as it is then. It was writing about more behavioral and psychology studies, but this is what it said. Typical studies involve two groups, one of which is subjected to some manipulation and the other of which is not. The mean scores of the groups are compared, and if they differ, it's concluded that manipulation caused the individuals to shift their opinions, their attitudes, or whatever. At least two things should be noted about this dreadful literature. That's his word, dreadful literature. First, between persons data are being used to make an inference of a within individual effect. And second, a group effect summed over persons is being used to infer a causal effect whose nexus is located within the individual. Neither inference is warranted. So that's a damning assessment of randomized controlled trials, but there is definitely a grain of truth there. So let's move on to why N of 1s. Next slide. So in the 19th and 20th century, there were two interesting developments. The developments of individuals was at the beginning and then groups at the end. But let's talk about some of the individuals. So Broca's aphasia. Broca was a French neurosurgeon who in 1861 heard about a patient with progressive aphasia with no change in their motor function. And at autopsy, he found a tumor in this area that he became called Broca's area and Broca's aphasia on a single patient discovered. And Fechner was a German philosopher and experimental psychologist who discovered the value and the meaning of two-point discrimination by testing himself. Freud, as you know, his work on the Oedipal complex came from a single case, Little Hans. He never even saw Little Hans, by the way, as you recall. This was letters from Little Hans's father to Freud. Ebbinghaus discovered the learning curve on himself. He was actually trying to, he was studying forgetting, and he learned about learning. Pavlov developed operant conditioning based on his own dog. And Norm Rosenthal, who the inventor of seasonal depression and light therapy, studied a single patient with light therapy, and that got into the news, and that exploded. And in fact, Norm Rosenthal himself discovered he had seasonal depression. So one person and one case can change our lives dramatically. Next slide. But also in the 19th and 20th century, there was also this time of growing of interest in the average, in the mean. So Quetelet's index is, Quetelet was studying what he believed was the human striving for the average. Does anyone know what Quetelet's index is actually called now? Alan might know. It's the BMI, body mass index, right? So it came from his belief that the humans strive for the average. Fisher, who is the grandfather or the great-grandfather of many of the statistical tests we use, including Fisher's exact test and the ANOVA, was studying the growth of grass blades. And so the whole basis of statistical tests in large groups is based on the growth of grass blades. And then large-scale randomized controlled trials eventually have become the highest level of evidence for directing clinical practice. And so this was the split. On the one hand, the value of the individual, on the other, the incredible emphasis on the value of studying groups. Next slide. However, there are problems with RCTs, and some are ethical. So we end up exposing large numbers of subjects to treatments that were either of uncertain efficacy or, in fact, no known efficacy, and that may be potentially unsafe. And some are practical. For example, we did a randomized controlled trial in adolescent depression, and we advertised for recruitment, and a week after we started advertising, the black box warning for SRIs came out. And so instead of recruiting 100 subjects per cell, we got 30 subjects per cell. That's the practical problem. And we exposed 60 youth to the treatment, and we found answers that were somewhat helpful, but a real many practical problems. There's a problem in averaging results, which was mentioned by Roa. And also, for example, in seasonal depression, when we started doing work in seasonal depression, half the patients who had weight change had an increase of weight with depression, and half the patient with a weight change had a decrease. So if you're looking at weight as a outcome measure, going up might mean you're getting better, and going down might mean you're getting better. And we have to average the results. So we lose the refinement of the data. And then there's problems with generalizability. So Bowden's classic study of Epival and the treatment and lithium in the treatment of bipolar disorder, they recruited 1 out of every 9, almost 10 patients they screened, they recruited. So this very influential study in the role of mood stabilizers in bipolar disorder really only looked at a very refined group of patients. So there are problems with RCTs that it's not individual enough. So for that reason, let's start talking about N of 1 studies. It's typical N of 1 studies in uncontrolled case history. First patient, when I was an intern, my first rotation was in psychiatry, partly why I ended up in psychiatry. And the first patient I ever treated was a woman who'd been on the ward for 6 months with a psychotic depression, and no matter what they did, ECT, medications, they couldn't get it better. So I'm this, green intern, really enthusiastic, come on the ward. I do every blood test known to man. Turns out she was profoundly B12 deficient, hadn't been tested. We gave her B12 injections, and within 2 weeks, she was discharged home with no change in her psychiatric treatment. And that was published in the British Journal. So the second kind of N of 1 study is a B design. And this is essentially, you do a measurement before you start treatment and you do a measurement after treatment. You know, there's all sorts of problems with it. I'm going to talk about mexiletine a bit later on. And then there's the, I guess the most common but still problematic design is the A-B design. So I'm really going through this to give you the nomenclature required for N of 1 study. So A means an observation period. No intervention is being done, and B refers to the intervention. So a sort of run-in period where you're doing measurement, and then the period of the intervention. I'm going to show you in a second why that's a problem, how that can be a problem. And so we get over it by taking multiple target measures and multiple time points. And I'm going to mention in a moment the statistical analysis for N of 1 studies. And also I'm going to talk to you about RCTs, how it's really important to get the question right and the analysis, and to be parsimonious with your analyses. Choose the most important analysis and don't do multiple tests because that affects power. But in the N of 1 study, we're allowed to actually transgress all those guidance and do as much as possible, as many times as possible. So next slide. I want you to just focus your attention on the right-hand side. And this shows change over an A and a B period. In the first one, in the top right, if we looked at the mean of that slope, we would say that treatment B worked, and the observation, treatment B was better than the observation period. But actually what's happening here is a trend across time that B may have nothing to do with. If we look in the second, if we look at the mean of those two slopes, A equals B, but we see a very different picture. During the A period, the person's getting worse. During the B period, they're getting better. But if we look at just one measure or the mean, we might find that they look the same. And similarly, in the last one on the bottom right, during the A, there was no change. There was a drop at the beginning of B, just before B, and then a clear improvement. But those means are the same. The slope is different. And this is why the recommendation is to do multiple measures across time in N of 1 studies. Okay, so what is an N of 1 study? Let's go back to our little nomenclature here. We talked about uncontrolled case history, the B design, the A-B design. I was going to give you an example of light therapy, we don't have time. And the third one, which is the most common one, is called the withdrawal design, ABA, a period of observation and intervention, followed by a period of observation. Now, this has virtually the same problems as the A-B design, maybe a little bit better, but many of those slope issues that we talked about can be problematic. There can be a crossover effect or a holdover effect to the treatment B, but unfortunately, this is what most people do. Next. So the next one I'm going to describe is the ABAB. This is called the equivalent time samples design, and it is essentially a gold standard. If you're going to design an N of 1 study, at least, and you have time and you can do it, at least do an ABAB design. It has problems, okay, but fewer than any of the previous designs, and fewer than the one that I'm just going to mention now, which is the BAB. This is a practical alternative. Sometimes you can't do an observation period, you just have to intervene, but at least intervene, withdraw, and intervene. It has the same problems as the withdrawal design, but it's better than nothing. And then you have multiple ABs. So AB, 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 AB. Yes, there is an advantage the more AB pairs you can do, but there's also now an ethical issue and a time delay and an effort and continual withdrawal for the patient. So, multiple ABs are great. You just have to be practical as well. If this was a lecture, or if we were sitting around a table, I might ask for a discussion about, examples you might have come in, but can't do it. There's also the ABCB design. I would ask you what you think that is, but I'm going to tell you. So A is the period of observation, B is one treatment, C is a different treatment, and the B is going back to the same treatment. So A is always a observation period, and then all letters after that refer to different treatments. So let's get a bit fancy now. In the next slide, I'm not going to go through all these, but you can now see before you design your N of 1 trial, one needs to be sure what you're talking about and can discuss it. I draw your attention to the second one, AB A prime to B A prime to B. The prime refers to a placebo. So in this design would be an observation period. B is the treatment. A is a placebo to B, then back to the treatment. Then A is another placebo, then back to B. And in the third one, B1, B2 refers to different dose levels of B, right? And the last one, I just put that up. That looks like I've just made that up and it's ridiculous. But actually, this was a study done at the NIMH quite a few years ago, in which the investigator took individual patients and gave them lithium, then Epival, then Tegretol. Then he gave them lithium and Tegretol, lithium and Epival, Epival and Tegretol, and then gave all three, lithium, Epival, and Tegretol. Quite a remarkable study demonstrating that the triple treatment was actually, and this is now you're looking at highly treatment resistant people. They had 1, 2, 3, 4, 5, 6 different trials before you got to ABC, and he still found around a 40% change. And he did this on sequential N of 1s. It wasn't a large scale trial. Every time a patient came up and was available, this is the design he did. Pardon me, next slide. I'm going to give you a spelling test now. So this is a 25-year-old male with newly diagnosed HIV. This was in 1987 when I was at St. Mike's. And he was started on azithromycin, which was the only treatment we had in 87. And about an hour after each dose, he had a panic attack. And so he refused to take the medication. So we searched the world literature, and there was no data on panic attacks due to AZT. And so we decided to go ahead with the N of 1 trial. Next slide. So over 24 hours, the patient got 4 doses. 2 were placebo, 2 were AZT, and they were randomly assigned. And the psych resident would assess 1 hour after each dose. So they were over 24 hours, so we had the resident on call do it, and I did it during the daytime hours. And what happened was, oh, see, it shows you there. Couldn't do this. Oh, there's supposed to be a progression. Never mind. The spelling test is here. The first randomly came as placebo, AZT, AZT, placebo, and that would be, well, you could call it ABBA, an ABBA design, a Mamma Mia design, or really it's the A prime B B A prime, or just to get a finer point on this, it's really B prime B B B prime, because the placebo is a placebo of B. It's not a placebo of the wash-in. It's a placebo of B. And you can imagine a study. We did a study once where we did light therapy and fluoxetine, and both had placebos. So fluoxetine would B, and the pill placebo B prime, and light therapy was C, and the sham light was C prime. So that's to put a finer point, but any one of those 3 would be acceptable spelling for that particular design. And just to show you what happened, he had 2 panic attacks during the 24 hours. Next slide. The first was after placebo, and the second was after AZT. And so we actually uncovered the blind to him. We demonstrated it was a random effect. He understood that and went on to take AZT successfully with no panic attacks, but actually went on to start to have panic attacks in the subway, panic attacks when he was leaving the home, and it was actually the onset of a panic disorder. Nothing to do with the AZT, had to be treated separately behaviorally, but he was willing to accept AZT. And, you know, in the old days, some of you on this call may not believe it, we used to have reprints. So people couldn't just copy it from the internet. They'd have to write to you as an author, and you'd send them a glossy reprint, you know. And I got more requests for reprints for this article than any of the large-scale randomized controlled trials or biological studies I ever did. Okay, so now I'm going to talk about how to apply an N of 1 trial, and I'm going to focus on drug studies. So next slide. Drug studies should still be double-blind if possible. I'm going to talk about what double-blind means momentarily. Yes, you have to worry about carryover effects. In the literature, it's suggested that drug carryover effects are more pronounced than behavioral. But I don't think that's true. I think, yeah, there's a biological crossover, carryover effect for medications. But CBT, for example, can work for months or years after you have a successful course, or mindfulness. So I don't know that it's particularly a problem with drug studies, but it is a problem. And then we talked about nomenclature. Okay, so some practical issues in the next slide. We have to select the correct time intervals and the number of treatment epochs. This is really important. As I mentioned before, you've got to show the slope rather than the mean. And it's okay to select multiple target measures, even though that's not acceptable in pure RCTs, and select statistics. So let's talk about statistics briefly. It is, I would say, apply both parametric and non-parametric statistics with caution, but with permission. I say it's a sin, but you will be forgiven. So here's the thing. In RCTs, we have to be absolutely super careful with how we use our statistics, with the parametric, non-parametric. In an N of 1 trial, it's really it's a true experiment, and it's true pilot. You can use whatever statistics you can reason, and you can do multiple tests on a single individual. If you're doing the A-B design or A'-B design, my suggestion is to do 3 cycles. I mentioned before multiple A-Bs are good, but at least 3 if you're looking at something like outcome, because by chance, half the people will get better on the first round, half on the second, half on the third. That means that after 3 trials, your P is less than 0.125. It's not quite 0.05, but in N of 1 circles, a p-value of less than 0.125 is still acceptable. Okay, so let's just talk about a couple of examples of when you can use N of 1. You can either a new or experimental treatment, or an application of an old treatment to a new situation, which I'm going to talk about shortly. If you want to ask, is the treatment really helping? I think it's helping, but I don't know. You can use the withdrawal design. Checking the origins of side effects, particularly in people with multiple treatments, multiple medications. It can be used for individual dose finding. It can be used for allergic desensitization and behavioral desensitization. So that guy that we gave the AZT, that's behavioral sensitization. And just yesterday, I had a discussion with a family and their family doctor about a young man who's getting severe side effects to vilazodone. And every other medication, and we're doing a behavioral desensitization using N of 1, which will start today. Or sometimes when you're stuck in a mindset, and I'm going to talk about that in a second. So next slide, and then next slide after that. So this is where we were stuck. So next slide. This is a woman, a 50-year-old female with severe treatment-resistant depression, and she has what Ken Schulman calls importuning. She was constantly seeking an answer to her absolutely unbearable anxiety, and she'd failed everything except she had a brief positive effect of Abilify once, and a brief positive effect of mirtazapine once. And she was currently on Cipralex with severe agitation, and she was determined to believe that the medication was going to work, and she refused to try anything else and move on, because she said, I'm sabotaging my own treatment. Please push forward with Cipralex. I know it's going to work. Next slide. So we did a 4-week trial, 7 days of Cipralex at 5 milligrams, that would be B1, and 7 days of Cipralex at 10 milligrams, that would be B2, and 14 days of placebo, I would call that B prime. You can also call it A prime. And she had to record her mood, anxiety, and agitation. And she also had a side effect of headache. And it was double blind. I didn't know what she was taking. She didn't know what she was taking. Only the pharmacist knew. And we revealed the results, and there was a very clear relationship between the agitation and the dose of Cipralex. On the day she got 5, she got some agitation. On the day she got 10, she got terrible agitation. She then had to believe it was the Cipralex, and we're able to move on. And we moved to a combination of Abilify and mirtazapine, the 2 medications that had a very, very minimal effect. That's 15 years ago. And she has been pristinely well 15 years, survived 2 bouts of breast cancer. I lost count at 8 grandchildren she now has. So she's done really well. And luckily we moved on from Cipralex. Okay. So the next is a new treatment. And I'm going to tell this is a story of mexiletine. In 1987, that was the bumpy year for N of 1 studies, right? So 1987, I was at St. Mike's and I got sent, New England Journal sent me a copy of that month's issue. And they said, there's more where this came from, please subscribe. I didn't subscribe, but I read that issue and I opened it. And there on this was an article of mexiletine. And in the yellow box there, you see it says mexiletine is a primary amine with close structural similarities to lidocaine. It has local anesthetic, anticonvulsant, and antiarrhythmic properties. I read that and I said to myself, those are the 3 properties of every successful mood stabilizer. Maybe mexiletine is a mood stabilizer. And so then next slide, I got Russell Joffe. I don't know why he agreed, but he agreed to treat the first patient in 1988. The 44-year-old woman who'd been hospitalized 15 times over 5 years with no well period at all. She was either manic or depressed for 5 years. Lithium had been effective, but exacerbated her terrible psoriasis, so lithium could not be used. So everyone agreed to a 2-week trial of mexiletine. It was going to be A-B-A. However, the trial was aborted because during the B phase, she completely recovered and was discharged home, and she remains on mexiletine to this day and is still stable. And over the next 10 years, we treated 10 individuals in the same ABA design and recruited 10 people. We published it. Ayel Schaefer came on board, and he then conducted a pilot RCT in 2000, which was his first 2 publications. It launched his career as well. No one else has done a study on mexiletine since. I still use mexiletine for bipolar disorder. I don't know if anyone else in the world does. Anyway, next slide. So, in summary, the study of one can help save lives, the lives of the many, right? Research careers can be built off the study of one. And N of 1 trials are simpler than you think, but I really encourage people to think carefully about its design. It's not, it shouldn't be an afterthought. It's got to be something you plan. There are multiple clinical and research applications of N of 1 methodology. I didn't even show you some of the studies I've done looking at biological measures in N of 1 studies that have been very instrumental. So let's start the revolution of the individual. I feel like Michelle and Michelle Obama there. Okay, so that's the discussion of N of 1. I'm now going to finish on, and do we have until 10? I want to stop at around 9:45 or so. Yeah. Okay, so I've talked about the value of N of 1. I'm now going to talk about the 3 rules of real estate: location, location, location. I'm going to talk about the 3 rules of RCTs: hypothesis, 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 sample size, and blinding. I was going to talk about feasibility versus pilot studies. We won't have time. So next slide, please. So the 2 most common fatal flaws in RCT applications is when there's a problem with a hypothesis or a problem with sample size. And you wouldn't think this would happen, but this happens frequently. So the problem with the hypothesis, it is sometimes stated in a non-testable way, or it's not even stated clearly, or sometimes it's not answering an important question. It's remarkable how many times there's a beautiful statement hypothesis, there's a beautiful data analysis, and the 2 are not joined at all. And so this is a problem of science. It's bad science if the hypothesis is not perfectly stated. And the second problem is with sample size. It could be unnecessarily large. That usually isn't so much a problem, but it's a cost issue and an ethical issue. But more often it's that there's not enough sample for power. In other words, it's a bad investment. If a CIHR or any agency is going to invest in an RCT, they want to invest, and usually they're very costly. They want to invest money in something with a testable question and you can actually trust the answer. So let's talk about these 2. Let's start with hypothesis. I say this so of course, you've always got to follow the clear headings and guidelines of the granting agency. I really don't like the headings for CIHR, randomized controlled trials. I really don't like it, but we're stuck with them. However, and I say this to experienced trialists, and I say this to new grads, all roads lead to and from the hypothesis. And it sounds like this is a parenthood and fruit dessert kind of statement, but it's not. Okay, there should be no extraneous information in the introduction or background or justification. Only that information which you are going to use to make a statement of hypothesis. No aims or objectives at the end of your justification, except those that serve the hypotheses. All inclusion and exclusion criteria have to serve the hypothesis. Now, that may sound like a strange thing to say, but I can't tell you how many times I've read a grant where there are 10 inclusion and 5 exclusion criteria, none of which are justified, and many of which it's hard to figure out how they relate to the testing the hypothesis. So as a way of training oneself, after each exclusion criteria or inclusion criteria, you say why. And the why has to be related to the thing you're testing. Okay, and then the next thing is only include tests, questionnaires, measures, investigations, or procedures that are about testing the hypothesis. Now, I put a little star there and a footnote. Yes, exploratory hypotheses are quite acceptable, but you need to distinguish them throughout the analysis, the sample size calculation, and even the budget. And I'll tell you why. I've read grants where an exploratory hypothesis involves doing MRI, and it's the most expensive thing in the whole grant. The grant might be a $500,000 grant. $300,000 is spent on the MRIs, and it's an exploratory hypothesis, right? So it's important that you realize that when you're asking for money, that's what you're doing. And I can tell you, CIHR will cut the $300,000 because it's an exploratory hypothesis. And then don't go and try and make it into a primary hypothesis just because you want to do it. Keep the hypothesis as it is. Okay. All data analysis is designed specifically to test the hypothesis. And I said this before, I've seen this example where the hypothesis and data analysis are disconnected. Every analytic strategy has to start with to test hypothesis 1, or to test hypothesis 1A, or to test the exploratory hypothesis. So it's very clear what your analytic strategy is testing, okay? And honestly, a study where the hypothesis and the analytic strategy, even though they're beautiful, don't match, will not fly, okay? It will die on the floor of the RCT committee room. And then at the end, when you're talking about what's the implications of a study, don't include any implication or conclusion other than what it means to accept or reject the hypothesis. I read grants where it's a MRI to measure resting state of a condition, and the conclusion is this is going to change the way we treat that condition. Well, that's a huge leap. It's not in the testing of the hypothesis. It's a mass. Tell us beforehand. Give us a warning. Otherwise, we get motion sickness. Tell us this is a massive leap. Okay, I accept that, but the conclusion can only be what you do if you reject or accept the hypothesis. Okay, enough off my hobby horse there, my little stand. So, next slide. Therefore, the hypothesis must be pristine, succinct, meaningful, and testable. It's a study that came through a couples therapy for major depression. Major depression affects relationships, and maybe if you do couples therapy, you can improve. And maybe the conflict in the relationship is the cause or the precipitant. So the design was escitalopram plus sham couples therapy versus escitalopram plus real couples therapy. It was an 8-week trial, pre and post. And the hypothesis was combined treatment will be more effective than either treatment alone. Again, it's so hard with Zoom calls, and then the test was analysis of various repeated measures with HAM-D, the outcome. The analysis is fine, but the hypothesis is problematic because it says combined treatment will be more effective than either treatment alone, but they weren't. The design did not include either treatment alone. It only included citalopram alone. You needed a real CT with a placebo citalopram, and you probably needed a double placebo. So in this, it shows that the investigator didn't fully understand the nature of their own trial. And great study, eventually got funded, not this time, we're not when it came to the committee this time, but this is a fatal flaw. And as I said, it will die on the floor of the room. Okay. The next thing I want to talk about after hypothesis is sample size. Incredibly boring topic, but I got to tell you, here's what traditionally I see 9 times out of 10, and I see it mostly in mental health and addiction studies, less so in surgery. So what I see is, what's the difference we expect based on our preliminary work or based on similar studies? What do we expect, or what difference would we like to see? And then a sample size is calculated on that. And that's a real problem because we tend to overestimate the effect. And the greater we claim the effect will be, the fewer subjects we claim we need. And the fewer subjects we claim we need, the less the power. So I'm going to show you this in an example, but let's just go to what's more acceptable than what we think we'll find. And that is a thing called the minimally clinically important difference, MCID. That is the smallest improvement that would be considered worthwhile. The smallest improvement that would essentially change treatment. And that depends on a whole lot of things, not just the scale, but what would change treatment. Now, sometimes we have no idea. We did a study in which the outcome measure was parental strain. And no one could tell us what reduction in parental strain is worthwhile. It definitely was the right outcome, but there was no literature on how to figure out what an important reduction in strain was. In those circumstances, you can use a thing called the Reliable Change Index, the RCI, which is the smallest difference that a scale or measurement can reliably detect. Okay, and you should calculate both really, but if you've got a good MCID, I'd go with the MCID. Next slide. So I'll give you an example of why MCID is important. So there's a new neuromodulation treatment for severe depression, severe treatment-resistant depression versus a placebo control. Now we know ECT results in a similar circumstance in a 20% difference between the placebo, 20% of people get better with sham ECT versus active treatment, 40% of people get better with ECT. And the goal is for the new treatment to replace ECT as an option. So if you say, this is what we expect to find, 20 versus 40, 20% placebo versus 40% new treatment, you need 82 per group, okay? But the new treatment doesn't need an anesthetic. It has fewer side effects. It works faster, doesn't require a visit to a hospital. And so when we ask patients and experts, what if there's just a 10% difference between the treatment and placebo? Would that change practice? And actually they said, yeah, we think it does. So that's the minimally clinically important difference. A 10% difference would change practice. So now we've got a placebo 20% versus a new treatment 30%. Now you need 294 per group. That's a whole different kettle of fish. And the sample size of 82 may have inadvertently said it wasn't an efficacious treatment. And we might have thrown out the baby with the bathwater. We might have said this new neuromodulation is useless. Let's move on to the next. But it's only because we based our sample size calculation on what we thought we'd find, not on a minimally clinically important difference. So let's skip the next slide. Let's go to the next. Okay, so there are problems with MCID. If we can, we'd base it on existing literature. But if we don't have that, let's create one. Survey people based on your hypothesis and your plain analytic strategy. Okay, don't forget that. And we include experts, not just experts that are part of the study, but experts beyond the study to say, what kind of parental strain reduction would be meaningful in your practice? What reduction in Hamilton score would be meaningful? What would change the way you practice if you saw it here? And if you can't do the MCID, then use the RCI, because the RCI is a psychometric calculation of the smallest reliable difference you can measure in a scale. And we just recently applied to CICHR, to the Randomized Controlled Trial, the Controlled Trials Committee, and we got all sorts of criticism. But the one thing that was mentioned by all reviewers is, we're so glad you used the RCI. It's so infrequently used. And this is because our outcome, there was no data on our outcome, so we use the RCI. So it's an acceptable thing to use. If you don't have it, you can survey patients as well as experts. So I'll give you an example. There was a study that was proposed, and it was a exercise regime to reduce the risk of cardiac events. And experts were surveyed, and they said a 25% reduction in risk would justify the wholesale adoption and change in this exercise routine. They then surveyed patients, and the patients said, we would never do that exercise regime. There is no percentage reduction that you can tell us. We're just not going to do it. And in fact, the study could not go forward. And very important information too. So I don't mind seeing, if I'm reviewing a grant, not just what the experts, but what patients tell us they would be willing to accept. And of course, you take the smallest of the two minimally clinical important differences to calculate the sample size. I suggest that people start with an anchor-based versus distribution-based MCID. What does that mean? Anchor-based is the number of points on the Hamilton scale, or the number of points on the PANSS, or the number of points on the Y box. And distribution-based is the number of standard deviation reductions, half a standard deviation reduction or a full standard deviation reduction. I'd start with anchor-based because it's more clinically meaningful. It's a number that people can digest more than they can a standard deviation, but I would accept either. And in fact, it's interesting. I've looked at some studies in OCD have used the distribution-based way of calculating a minimally clinically important difference, and it's acceptable. And the last thing I want to say is, if you have a hypothesis that has both improvement and a worsening, you've got to decide which is primary. For example, studies in bipolar disorder, the main hypothesis can be improvement in symptoms of mania. The secondary hypothesis can be no rapid cycling or no induction. So improvement in symptoms of depression, and the second can be no induction of mania. And those two are maybe in opposite directions. So I just would choose whichever's primary if you can. Okay. So the last thing I want to say before I open up for questions and discussion is about blinding. Next slide. So next slide, please. Sorry. So blinding is all about who didn't know, what they didn't know, and how do you know they didn't find out, right? We talk about double-blind studies. We're now talking about triple-blind studies. Traditionally, triple-blind is the patient is blind, the assessors are blind, and the statistician doing the analysis is blind. But actually, there's more people that need to be blind. And I don't know when people say double or triple, what they're talking about, okay? So I want, what I would prefer, and this is Lang wrote this almost 15 years ago. I just want to know what's blinded, okay? Who didn't know, what they didn't know, and how do you know they didn't find out? I'll show you the next table. Next slide, it's a bit dense here, or small, but essentially this is a table that needs to be reproduced for every study. And it says that on the first column is group or individual blinded. What was the information that was withheld is the next column, and the final column is the method of blind. So that the third column is method of blinding, and the final column was, did the blinding get compromised? And that's what I need to know. And you can see here, the person assigning the participants themselves, the care providers, the data collectors, the outcome assessors, and the statistician. There's actually 6 groups of people. Plus, beneath that, you see the trial manager, the pharmacist, the lab technicians, if it's necessary, the safety committee, and the manuscript writers. There's multiple ways that you can be blinded. And I just rather see this table reproduced. Tell us who's blinded. And I'd like to also see, did you check that they were actually blinded? I want to just briefly talk about the manuscript writers. So Rula Markoulakis, my colleague, and I, in the process of publishing a randomized controlled trial. And we went through the exercise of writing out our discussion So we had the groups were blind, the patients, the subjects were blinded, the assessors were blinded, the statistician was blinded, and the statistician just gave us group 1 and group 2. That's all we knew. And Rula and I went through the exercise of writing the conclusion based on group 1 having more significance or group 2 having the more significant findings. So we compelled ourselves to make comments either way before we uncovered the blind. That's quadruple blind, or I don't really care about the numbers anymore. We were manuscript writers who were blind to the assignment when we wrote the conclusions. Okay, last slide. So I hope I've given you a sort of a taste of the value of NM1, both for patients, for the general scientific community, for society as a whole, and even for careers. I've spoken about a few guiding principles for randomized controlled trials. Please focus on the hypothesis. No more sample size calculated on what you think you'll find. Please use MCID, and don't just count the blinding, single, triple, quadruple, whatever. Tell us who is blinded. And thus endeth my presentation. Happy to accept questions.